Hello, good morning, good afternoon, good evening, depending on where you are in the world. My name is Im Oliveras. I am one of the part of the steering committee of the climate change thematic group of ATBC. We are a group interested in climate change impacts in tropical systems, and anyone a member of ATBC is very welcome to join the group. We organize usually activities. Last year, we organized a series of webinars, and this year we are organizing this symposium on climate change effects on species interactions in the tropics. So if you're interested in joining, just drop an email to someone in ATBC or to myself, and we would be very happy to include you as part of the group. So during this symposium, you may submit questions to the speakers and at any time during the presentations, but please use the Q&A function on Zoom. Don't use the one in WOBA. And if you are addressing to a particular speaker, please indicate the name of the speaker. So today's speakers are Rob Badji from the University of Connecticut, and then Adriana Corrales from the University of Rosario, and Karine Emmer from Universidade Federal de Pernambuco. So we will now begin the presentations.
Hello. Today, I'm going to talk about how natural enemies mediate species coexistence in tropical forest tree communities and how those coexistence mechanisms might be affected by climate change. Now, we all know that tropical rainforests have exceptionally species-rich plant communities, and there is a huge literature that examines what mechanisms might contribute to those patterns. And among those mechanisms, one of the most talked about is this one here, which is often referred to as negative conspecific density dependence. And the way this works is when a plant species becomes particularly abundant, the insects and pathogens that attack that species are drawn to this concentration of resources that increases the mortality of that plant species, which means that there is space for other species to move into. One of the critical components of this hypothesis is that the natural enemies need to be relatively host specific so that they don't spill over onto rarer species when they're attracted to high concentrations of their hosts. Another mechanism that has been discussed in the context of tropical forests are storage effects. And the way these work is that if you have different species fruiting and germination phenologies being separated in time, then different species won't be abundant at the same time. If insects and pathogens are attracted to a high concentration of one species, they will have affected high mortality in that plant species before another species, perhaps a rarer or less competitive one, germinates. And so that later species will not be impacted to the same extent by either competition or natural enemies that are attacking the first species. A critical component of this hypothesis is that there needs to be a relatively invulnerable stage during which plant species are not affected too much by either competition or natural enemies. And in the case of tropical trees, the adult phase would fit the bill. An important component also is that even generalist natural enemies can cause species coexistence under this mechanism. And to summarize these two mechanisms, they both involve a barrier to the exchange of natural enemies between seedlings of different plant species. In the case of negative conspecific density dependence, that barrier is the host specificity of their natural enemies. In the case of storage effects, the barrier is different fruiting and germination phenologies. Now, how might this all be affected by climate change? Well, we know from looking at general circulation model outputs that there are going to be shifts in precipitation patterns through the tropics. In the neotropics, we expect that by the end of the 21st century, South America and Central America are going to become relatively drier than they are at the moment. Throughout the tropics, for the most part, we're also going to see increased seasonality in large parts of the tropics. So the question that I'm going to start off by addressing is, to what extent annual precipitation influences the strength of negative conspecific density dependence? Do we see that as precipitation increases, negative conspecific density dependence increases? In which case, if precipitation declines in the neotropics, we might expect to see a decline in the strength of this mechanism, which could erode species richness in the tropics over the long term. And there is some indirect evidence for this idea. If we look at a regional scale, plant species richness increases with mean annual precipitation. And a little bit more directly, a meta-analysis by Liza Komida and colleagues conducted a few years ago suggested that studies of negative density dependence conducted in wetter regions tended to detect stronger patterns than those conducted in drier forests. So there was an increase in the strength of negative conspecific density dependence with the precipitation of the site at which the experiment was conducted. In order to look at this within the context of one study, with some colleagues, we set up an experiment across the Isthmus of Panama, which is associated with a strong precipitation gradient. And at each site, we measured the strength of negative conspecific density dependence across the seed to seedling transition. And we also treated plots with insecticides and fungicides in order to determine whether these natural enemies might be causing negative conspecific density dependence and any change with the precipitation of it. And when we looked at the results of this experiment, we found that there was evidence of negative conspecific density dependence, but that effect did not seem to vary with mean annual precipitation. It was pretty constant across the isthmus. Furthermore, there didn't seem to be any evidence that either fungi or insects were driving this negative conspecific density dependence. There was no effect of insecticides or fungicides on the strength of negative conspecific density dependence. I should caution that these are relatively preliminary results because I'm still in the process of taking a deep dive into analyzing these data. We can get at density dependence at a later stage of plant development by looking at spatial patterns. We'd expect that if negative density dependence is going on, then trees should become less clustered as they age, because dense clusters of seedlings and trees are going to suffer higher mortality. And if precipitation is correlated with stronger density dependence, we expect a larger decrease in clustering at wet sites than dry sites. However, across the Panamanian Isthmus at least, we actually see that that decrease in clustering between small trees and large trees at the drier sites, but at sites with a milder dry season, which is how I measured precipitation or how wet a site was in this experiment. At sites with a milder dry season, we actually saw a smaller decrease in clustering between small and large trees. And to look at that a little bit more directly, I've plotted the change in clustering between small and large trees here, again, for both site with a dry season and a mild dry season. And you can see that the dry season site, when in yellow, has a greater decrease in clustering over the small to large tree transition than the site with the milder dry season. To hear more about our work on density dependence and pathogen attack across the Isthmus of Panama, I urge you to go to a talk by my PhD student, Val Malisi, where she will talk about pathogen incidence rates across the Isthmus of Panama, and that talk is later today. So, in response to our question about whether negative conspecific density dependence seems to strengthen as annual rainfall increases, perhaps not, at least according to the data that I presented today. So, on to our second mechanism. Could storage effects decrease as seasonality increases? And the way in which this could work is, as seasonality increases, plant germination patterns are concentrated within a smaller window when conditions are good enough for germination. If it's really dry, plants will have a hard time to actually survive during that initial germination period. And so that could concentrate plant phenologies into a smaller window, and therefore they would overlap more, decreasing the potential for storage. And I haven't done any work on this yet, but if we look at a paper by Jacob Itsinovitz and colleagues that came out a few years ago, we see that as seasonality increases, the potential for storage effects to cause species coexistence actually decreases. Okay, so more aseasonal climates allow for greater separation of tree fruiting phenologies, which allows for greater species coexistence via storage effect. So in summary, therefore, we expect that climate change will alter precipitation patterns throughout the tropics, and it seems that negative conspecific density dependence is unaffected by mean annual precipitation. So at least according to these data, it doesn't seem like climate change should affect this particular mechanism. However, increased seasonality, which is expected across a lot of the tropics, could disrupt coexistence via storage effects. And with that, I'd like to thank you and my colleagues who have contributed to this work, and draw your attention to a paper that we wrote a year ago that goes into more detail in some of these ideas. Thank you very much for your time.
For this invitation to talk today about the effects of climate change and mycorrhizal neutralisms in tropical forests. What are the questions we need to tackle? So the rhizosphere is considered a hotspot of microbial activity in the soil because it hosts a highly diverse community of microorganisms and intervene in biogeochemical cycling, influences plant community diversity and composition, and shows a lot of multi-trophic interactions. One of the most important functional groups in the rhizosphere are mycorrhizal fungi. Mycorrhizal fungi form symbiotic associations with the roots of plants where fungi transfer soil nutrients to the plant in exchange for sugar. The most important groups of mycorrhizal associations are arbuscular mycorrhizas, ectomycorrhizas, ericoid mycorrhizas, and orchid mycorrhizas, and they all differ in their biology and ecology and functional traits. For example, arbuscular mycorrhizas specialize in phosphorus acquisitions, have lower carbon costs for the plant, have limited access to complex organic substrates due to low enzymatic capabilities, while ectomycorrhizas specialize in nitrogen acquisition, have higher costs for the plant, and are able to break down complex organic substrates due to high enzymatic capabilities. The effect of climate change on plant mycorrhizal interactions could be direct by changes in abiotic conditions like temperature, precipitation, and nutrient availability. These changes can have direct effect on plants or fungal ecophysiology, or could be indirect, mostly driven by changes in species functional traits and phenology, shifts in species distributional ranges, and changes in species interactions. These climate change effects can result in altered feedbacks between plant and fungi that can affect ecosystem processes. Recent studies have shown that interactions and complementary effects of climate change factors could significantly alter ecosystem processes at faster rates than predicted if studied individually. Also, fungal functional roles could be context-dependent, and therefore, future species interactions under climate change conditions could be hard to predict. It is well known that climate change produces shifts in species altitudinal distribution, with mountain species migrating upslope in the mountains. In addition, a recent study demonstrated that tropical trees are migrating upslope 10 times faster than their temperate counterparts. However, it is unknown how the root-associated fungal communities will respond to shifts in their host distributional range. To improve our knowledge about the current distribution of mycorrhizal associations in tropical mountains, we wanted to understand how climatic and soil conditions shape the distribution of mycorrhizal types along altitudinal gradients in the Colombian Andes. So we use more than 260,000 herbarium plant records and bioclimatic and soil variables at a resolution of 10 kilometers to answer this question. We found that plants with different mycorrhizal types differ in their altitudinal ranges, with arbuscular mycorrhizal and nitrogen-fixing plants associating to lower elevations, while ericoid mycorrhizas and orchid mycorrhizas associated to higher elevation. We also found that arbuscular mycorrhizal plants show a positive association with temperature and negative associations with soil total nitrogen, while ericoid mycorrhizal plants show negative associations with temperature and positive associations with total nitrogen. With this study, we concluded that climatic and edaphic variables have the highest explanatory power for the distribution of different types of mycorrhizal associations in the Colombian Andes. Therefore, changes in climatic conditions can strongly alter these distributions in the future. Potential climate change outcomes could differ among plants and fungal species, depending on their dispersal capabilities, with species with higher dispersal capabilities being able to migrate faster than species that experience dispersal limitations. Also, plant distributional shifts will affect their interactions with mycorrhizal fungi, leading to partner mismatches and interaction losses, depending on the amount of inoculum availability. In addition, mycorrhizal species could migrate deeper in the soil profile, causing interaction losses as well due to separation from their host from the host roots in the soil profile. So, what are the main knowledge gaps that, from my point of view, should be filled to improve our understanding of plant mycorrhizal associations in tropical forests? First, I think it is important to improve our understanding of the species composition of root-associated fungal communities in tropical ecosystems. Metagenomic studies from soil communities have shown that tropical fungal communities have higher species richness and that many of the species are undescribed. This makes it more difficult to define distributional ranges for a species. In addition, it is important to understand how fungal dispersal capabilities might affect plant dispersal limitation when colonizing new habitats due to inoculum limitation. In addition, it is important to understand how mycorrhizal fungi with contrasting functional traits respond differently to climate change and alter differently ecosystem function. And it is important also to find associated trade-offs for root-associated fungal communities. Finally, I think it is important to document the degree of host dependency on mycorrhizal fungi and how these mycorrhizal fungi contribute to plant fitness under different environmental conditions. So moving forward, from my point of view, I think we should focus on three main aspects. First, we need to initiate coordinated efforts to establish replicated studies across the tropics to try to find patterns in all tropical forests. Second, we need to leverage the use of omics, metagenomics, metatranscriptomics, proteomic methods to improve our understanding of community composition and their associated function. Finally, we need to increase the use of experimental approaches under controlled conditions, combining multiple global changes and associating those changes to ecosystem function. Thank you so much for listening, and I'm ready to take any questions.
Hello, everyone. Thanks a lot for having me here. It's a real pleasure to be able to talk to you a bit about my research line, and I hope this finds you well and safe during these difficult times. So I'd like to start by acknowledging the title of my presentation, which is related to this book of Hutchinson, which I read during my undergrad studies many years ago, in which I think it describes very well the way that I see and the way that I analyze how humans are affecting the way species interact. So the idea here is that we have this ecological time where species coexist and they interact in different ways, of course. And the evolution here is not only what brings the species to this ecological time, but also a consequence of what's going on in this ecological arena. So the way that species are interacting now and the factors that are involved within the ecological time will also affect the evolution from now on. So with that in mind, I would like to introduce you the different components of our ecological tier and the evolutionary arena, which are the actors to start with, which are birds and plants from the tropics. And what they are doing in this theater is seed dispersal. So seed dispersal is a mutualistic association between animals and plants, fundamental to maintain the functioning of forests, especially in the tropics, where we can have up to 90% of the trees relying on animals to be dispersed. So which means that when we look to a beautiful forest such as this one, which is actually one of our field sites, we are seeing not only trees, but also the interactions involved on those trees. In this case, we focus on seed dispersal, so which means that each of these plants are producing fruits that will be once consumed by different animals that will disperse them, hopefully to safety sites where they will be able to germinate and grow and hopefully succeed. And also they are a consequence of some event of seed dispersal from which they are able to be moved to a safety site and grow and get to this mature stage. So in this scenario, we add the Anthropocene, which is the area in which we are living, where we have almost half of the planet already transformed by human activities. Oops, sorry about that. And here we get to our theater, which is the Atlantic forest of Brazil, in which we have around 12% of the original cover remaining only. In this scenario, we still have some of these larger areas, but most of the area is placed in very small fragments surrounded by different types of matrix associated to defaunation, which is the loss of species and populations. So what does it mean? It means for our system that this is what we expect to see if we go to a pristine forest. But when we go to a defaunated area, this is what we would see. So a reduced subset of sizes, functions, and evolutionary history, as I'm going to show you. Spoiler. So our play is composed of different scales. So we want to know what's going on to see dispersal at the local scale, regional scale, and also at this eco-evolutionary time. I'm presenting to you the main results of 3 papers that came from my postdoc. As I don't have much time, I will just give you a brief summary, and I would like to invite you to read the papers if you like, or contact me later to discuss any of these ideas. So when we look at the local scale, what we do is go to these fragments and record the interactions that are there between plants in blue and birds in orange, and then interactions among them in gray. And then you can see from 1 to 16 that as the disturbance increase, the structure of these networks are changing as well. So even if you don't understand anything about networks, you have to agree that 1 is different than 16. A number of nodes, they have number of interactions, and the way that these interactions are organized within the network. So it means that the complexity of the system is changing. Then we move from the local to the regional scale, and we use something that we call the meta-network approach, from which we scale up, and we are interested on the connections among fragments, here represented by these blue lines, which are performed by an association between a bird and a plant species that have with them the different traits, like body mass and seed diameter, that are the ones we use here. So this is the structure of the meta-network in the Atlantic forest. So it's highly modular. So each of the fragments correspond to a single module in the system. So these clouds of nodes. Just looking a bit up, so we have the squares representing the fragments and the local interactions, so those ones that occur in a single fragment only. And then we have these interactions that are actually occurring more than one fragment. So those are the network connectors, so the blue lines that I show you in the figure just before. These are important because those are the ones responsible for connecting the landscape. If we do this exercise of removing them, as in C, we see the fragments get completely separated. And we don't want that because we are losing then the flow of information between fragments that will then reduce genetic diversity in a middle to long term. Another important point here to call your attention is the effect of area in these interactions. So we have these functional groups here represented by size of birds and size of the seeds. And it's important to note that the purple ones, that the small associations, are present throughout the whole gradient of area. And the other group, the large interactions, that are ones that will be related to a higher capacity of carbon storage as well of the plants, need much larger areas to start to appear in the system. Then the last part I would like to show you is about the evolution. So we are interested on how many millions of evolution we are losing in this scenario. And what we did here, we want to know the how many years of evolution of the interactions. And the way we do that is to get our associations between birds and plants and bring the phylogeny of both groups to the equation. Then we calculate an index that gives us an idea of how unique each of these interactions is within their own phylogenetic group. And then we just sum the index, the ED, as we call it, for the of the birds and the plants that interact. So we get an index to the interaction level. Then we make some associations again with body mass and seed diameter, and we see that larger seeds and larger birds are more unique within their phylogenetic group. And then when we put them together in the gradient of defaunation, what we see here, without going to further details of what each of these associations mean, what we can clearly see that is, as we increase defaunation, we have this drastic reduction in the proportion of the interactions that remain in the system. Then we can ask how much evolutionary history each fragment holds. And again, in the gradient of defaunation, the numbers here correspond to each of the study sites in the system. Number one is the most pristine, and the larger the number, the increase in the defaunation, the disturbance in the area. So again, as we move in the gradient, we already see a drastic loss of evolutionary information in these anthropogenic landscapes. Right, so what we identified are mainly two groups, vanishing and persistent interactions, and we can see who they are, so their identity, the traits that they bring, the functions they perform in the system, and as well as the evolution associated to that. So finally, what does it have to do with climate change? I would say that species interactions are either the drivers and the consequences of climate change, and we can think that in different ways. Just an example of questions that are remaining open for further research is we can think about how much carbon storage we are losing with the loss of nature's complexity, or we can ask the other way around, how increasing carbon loss is affecting nature's complexity. And then we can take at that in different scales likewise. So we can take at that at the local scale, at the regional scale, and how this is can have consequences in the evolutionary dynamics. With that, I would like to end acknowledging my collaborators and thanking my sponsors. And of course, thanking you for your attention and for, again, thanking the organizers for the great opportunity to talk to you today.
Great. Well, thank you very much, Corinne and Adriana and Robbie, for some really excellent talks. We now have an opportunity to have a question and answer session with our speakers. So if you'd like to raise some questions, please do enter those using the Q&A button on Zoom. And my name is Jim Dowling. I'm one of the moderators, and so we'll have about 15 minutes of discussion, I think, before our session is due to close at 9:20 my time, at least. And so Arildo Diaz, who is the other moderator today, will also sort of summarize some of our discussion as well at the end of this session. And also just to note, we've added on the Hoover, if that's how it's pronounced, webpage, a link there that you can continue to add your questions, and we can continue some discussion of this theme. And so sort of two components of our discussion today. Hopefully, one of them, are the direct questions that we have for our speakers about their talks, but we're also interested in mediating or developing some ideas for developing research programs to detect climate change phenomena. So your ideas for potential case studies or systems that could be used to investigate across different sites or potentially spatial scales. So I'm going to start by looking through our question and answer session. And actually our first question, which came from Sean, is related to this issue, and it's specifically for Robert. So at what spatial scale are trends in NCDD, negative conspecific density dependence, observable? What scales must we work to better predict the effects of climate change on the changes in spatial associations?
I think that's a fantastic question, and it is a question that we all end up making arbitrary decisions as to what scale we're going to study because it's what's practical. And that, of course, makes it harder to generalize. I think, we're talking about two different spatial scales, or two different processes that we need to think about the spatial scale of. One is the spatial scale of density influencing an individual plant's performance, survival or growth or reproduction. And that, I think, we do have a relatively good answer to, which is about maybe 15 to 20 meters away from the individual plant seems to be the spatial scale at which natural enemies or whatever processes are affecting survival through density are acting, whether that's because of the spatial patterning of fungal communities or insect communities. We do seem to have an answer there. Now, the harder question is, at what scale would things like climate, at what spatial scale is climatic variation influencing the strength of any sort of negative conspecific density dependence? And that is a difficult question because generally the scales we've chosen to study are completely arbitrary. It depends on what data are available. And at the same time as we're seeing variation in climate in terms of precipitation or say temperature, there are also other things that are co-varying, soil properties or biogeographic regions, etc. And I think that one of the interesting contrasts between Liza Comida's study and the work I presented from Panama is Liza's work was a meta-analysis. It was taking data from right across the world. It was temperate and tropical, And so therefore, you're getting a very large spatial scale of variation in climate, whereas the work that we did in Panama was over the scale of 120 kilometers. Admittedly, over, we had a very strong precipitation gradient that we were working on, but it's a more limited spatial scale. And so, I don't have a straightforward answer, but I think in terms of the theme of this session, what do we do next? I think we need to try and find as much variation in climate while keeping other things as constant as possible. So, if we're shifting biogeographic realm and we're introducing species that have completely different evolutionary histories, species interactions that have completely different evolutionary histories, then there's a lot of confounding factors. And so a smaller spatial scale and as much variation is probably what we want to work on. So long-winded way of saying it depends, I guess.
Thanks, Robbie. I'm actually going to follow up with one more question for Robbie before we move themes a little bit. This one comes from Camilla, and it kind of is another aspect of scale, I think. So one issue I think we have in doing experiments that address issues of climate change is, are the species that we choose somehow representative of their communities? So there's this issue of sort of idiosyncrasy, which results from just not having a very large sample of species. And so Camilla's questions are more related to sort of the details of your field experiments. So how many and which plant species are you using? How long do you run an experiment for? What kind of genetic information do you include? How do you deal with the below ground organisms interacting with plants in your experiment?
All excellent points. So I'll try and answer the simple ones first. The experiment, we did not try to manipulate the species that were getting into the experiment. So it was basically the natural seed rain and germination patterns that we were looking at. That said, there was between 12 and 20 species at each site that I could use in those analyses that I presented today. There are, there's about 200 species in the data set, but in order to get at NCDE patterns, negative conspecific density dependent patterns, you need enough variation in seed numbers to actually be able to fit a line through the regression. If you have no variation, then you can't see how survival is varying with it. So that's a tough constraint. In terms so the experiment was run, for 18 months. And so actually it's closer to 2 years. So ideally you'd have a longer run because you get more data. And some of the data that Joe Wright is collecting from Panama, for example, and a number of other sites have similar data, is much longer. And of course that gives you more power. The problem is applying pesticides for more than about 2 years becomes a little unwieldy, just because it's every 2 weeks you're having to send someone out to the field for a full day at 8 sites. That starts to get to be a lot of work. In terms of below ground processes, I mean, to some extent, below ground pathogens would be suppressed a little bit by the fungicides that were being applied, but it's likely to be relatively small. Most of these effects are going to be above ground interactions, and that's because fungicides are mostly travel up the plant. They are most manufactured fungicides are transported in the xylem, not the phloem, and so therefore below ground processes are not modified to the same extent as above ground processes. We wanted to actually unpick that a little bit this year, but there's this virus that's going around that kind of impeded our plans, so that didn't happen for reasons that we're all aware of. So hopefully that answers your question.
Great, thanks very much, Robbie. And so we're going to pass along to Adriana now. So there's a question here from Yit, which is how common is the fungal or mycorrhizal ability to change roles? So the flexibility the functional role of fungi? Is this something restricted to a small group of taxa, or is this something more general, or something we don't really know anything about?
Yeah, it's a great question. And yeah, thank you for that. I think we don't really know what is the proportion, because for mycorrhizal itself, I think it's more like variation in the benefits for the plants, like how much carbon is costing for the amount of nutrients that the plant is receiving. So in that sense, it could be still working as a mycorrhiza, but transferring less nutrients to the plant than the carbon that is costing. So it could become like a parasite in the spectrum of the parasitism. And that's very hard to measure, because you have to measure the fitness of the plant and the amount of colonization and the amount of carbon transfer and etc. So that's a little bit difficult to quantify. So I think we don't know that aspect. There are other fungi that could be, for example, root endophytes, that could also behave like mycorrhizal, that could be mutualists, but under certain conditions or different hosts could become pathogens too. So that's why it becomes too complicated, because it's host specificity and this idea of functional specificity that Jim, Camilo, and Salamea and Carolina have worked on, which is so interesting. It depends on the host, it depends on the environment. For example, the soil, the amount of nutrients in the soil. So it's very complex, and I don't have a clear answer for that question, but it's a great question. That's the type of questions that we have to start answering. Thank you.
Thanks very much, Adriana. I think you kind of answered Alexander's question at the same time, so that was very efficient. So, but there is another one here. We may just follow up a little bit more on the issue of soil. So Akshay was asking about the fact that mycorrhizae can track nutrients, and that we know that climate change is altering decomposition. And this interaction in the soil between decomposers and mycorrhizae are very important in determining soil carbon storage. So do we have any predictions for how decomposers and mutualists might interact with rising temperature or respiration rates? That's a tricky question, actually. It's a good one.
Yeah. I think that question is right at the frontier right now. I don't think that's what everyone is trying to answer right now, because yeah, also like soil nutrients affect plant composition and microbial composition in the soil, fungal composition. So the composition can change from soil types or when you add nutrients, but that also can change with temperature, and the biotic interactions between microbe to microbe, microbe to plant will change as well with nutrient addition and temperature and water as well. So it's very tricky, because we know that, for example, ectomycorrhizals can decrease inorganic nitrogen availability in the soil. That can slow down decomposition, at least in some systems. That's the opposite for arbuscular mycorrhizas that are more abundant when there are more nitrogen availability, that can increase decomposition. Now, when we have both types of mycorrhizas, which one is going to win, which one is not going to win, and what's going to happen with nutrient addition is just, it's very complicated. I don't have a clean answer for that one either. Sorry. But it's a very good question.
Thanks, Adriana. So, Karine, we've got a couple of questions for you. And actually, maybe the one that Sean asked is most relevant to you. So maybe you'd like to start off with this. So Sean says, super interesting presentations, thank you. While protected areas can protect species refugia under climate change, what can we do to mitigate these shifts in conspecific and interspecific interactions?
While protected areas can protect species refugia under climate change, but can mitigate these shifts. Woo, that's hard.
No easy
Well,
questions in this session.
no easy question. Yeah, I'm not sure I understood exactly what you mentioned, but from our study, what we see from the Atlantic forest is that I haven't showed much, but is that we see the remaining fragments that are not necessarily the protected ones show different interactions than those in the protected ones. So I think it would depend of the scenario that you have. So if you have a scenario like the Atlantic forest that's already highly degraded, you might need to conserve or protect all the areas that are left, because you see a huge beta diversity of species and interactions, which means that as you go to different areas, you find different aspects of biodiversity there that are these refugia of species, of evolutionary history, of genetic information, and so on. But if you go to different areas like the Amazon, for example, the answer might be a bit different, because you still have large areas of continuous forest, and the ways of protecting those areas and to mitigate these chains on species interaction might be a bit different, and perhaps a bit higher, more complex, I would say, as we don't really have much information from those areas. So. Yeah, I think that's the point I would like to make, really. It depends on the context. Hard to say.
Thanks, Corinne. Yeah, I mean, this has sort of occurred to me as you were giving your talk. You know, so Robbie has sort of emphasized the value of using these long gradients of precipitation, or directly looking at environmental variables. But we also have in communities gradients of fragmentation and disturbance, which has resulted in species loss in a way which is sort of comparable to what we're predicting about how community simplification might result from climate change. So maybe we actually have two axes that we could bring to bear in terms of thinking about using natural communities presently to predict or project climate change effects. So I don't know if Adriana or Robbie or Corinne want to follow up on the original question and think a bit more about how we integrate fragmentation and disturbance as a model system for understanding how climate change might influence more continuous ecosystems, or less fragmented or less disturbed ecosystems.
Can I just compliment then in these? Yeah, I think I try to make a point in my talk. It's more like a provoking than giving an answer, but is that the way that's actually, I was talking about species interactions, but it can be fragmentation as well, or deformation that were the drivers I analyzed. So those as either drivers of climate change or consequences of climate change, I would say. So you can think about, there is a colleague of us that made some modeling about the loss of the larger body mass birds that disperse the plant species that have larger seeds, and those ones are the ones that are known to storage more carbon. So if you lose these interactions, because you have habitat loss, you have hunting and many other drivers, you might have a consequence in the middle to long term to the carbon storage that this forest or these fragments can hold. This is a study from Carolina Bello, I forgot to say. And there is another study from a colleague, that Lilian Salis, that were modeling the opposite. So she was modeling the distribution of plants. So how the change in the distribution of plants can influence the species interaction in the long term. So maybe we can go to that way as well. The point is that many of these climate change modeling are not able, as far as I know, to include species interactions that are the base of this architecture of biodiversity, as we say when analyzing ecological networks. And it's not trivial to include them. So maybe this is a point also to consider.
Yeah, I don't know, Robbie or Adriana, if you have any thoughts to sort of add to this.
Maybe I can add something. I mean, I don't know the literature, but I think it would be very important for example, for the mountains, we need to protect mountains because are gonna be where the plants are gonna migrate to, and if the tropical plants are migrating 10 times faster than the temperate plants, then we definitely need to protect mountains to be able to allow those migrations to occur for both plants and insects, fungi, birds, like all the associated interacting groups. And also, I think it would be very important to really understand. I think about, for example, the systems that I like to study, which are these tropical ectomycorrhizal monodominant forests, which are relict species with high density, but they are very rare and they are all pretty much endangered species. And they are mostly from mountain forests. So we need to try to predict how far they are gonna migrate in the next, I don't know, century or something, and try to protect those areas because otherwise they're gonna disappear.
I can jump in if that would be useful. So, I mean, my perspective on this is that we're dealing with really, really hard questions, right? Because we're trying to project, we're trying to make predictions about situations that don't currently exist, but unfortunately will exist in the future. And I think the only way to make those projections is, you know, a synthesis of lots of different approaches. And I think, you know, it's why collaboration is so important. But we need the experimental studies to work out what the interactions are. You know, the kind of work that Corinne and Adriana have been talking about, and my work as well. You know, the small scale work to really work out how the mechanisms are working. But then we need to expand that with observational studies to see how that's varying over spatial gradients that we cannot manipulate. And then the only way to pull all these things together is development of theory and data intensive analyses. And that sort of speaking across techniques and disciplines is so essential if we're going to address these problems. And unfortunately, that's really hard because you know, it takes depth of experience and depth of knowledge in each field, but also an ability to collaborate across fields and knowing enough about. So the breadth and depth combination is always hard. And the only way to do that I can imagine is collaboration. And so, you know, I think symposia like this are really helpful because they force us to think at those spatial scales.
All right, I just want to ask one more quick question before I pass it across to Rildo. And so at the same time we're trying to understand and predict the effects of climate change, we also need to be thinking about mitigating the effects as well. So we're essentially developing management at the same time as we're understanding the problem that we're managing. And so there's a follow-up question for you, Corinne, here about do you believe restoration plantings are likely to restore plant-frugivore interactions in Atlantic forest fragments, or just more generally, you know, is this sort of restoration planting approaches that have been promoted, including sort of predicting where species are going to be under future climate scenarios and providing, you know, the interactions that might be lost otherwise by management is an approach that you would advocate, or maybe particularly in the context of the Atlantic forest that you're working in?
Yeah, difficult question.
Not really a quick question. Quick answer,
a whole
maybe.
day to think about the answer. Yeah, of course. I mean, of course, restoration is really important, but there's these huge discussions within the restoration field is about what to restore and how to restore, right? So it's not, again, it's not easy to say, but yes, there are some discussions and studies and initiatives also to restore interactions. It's still, I mean, it's very, it's just at the beginning, but it's a very important point also, how to restore ecological interactions. because we need to restore both, right? So we don't want to just plant trees if you don't have the animals there, or the other way around. You don't want to just reintroduce animals, for example, rewilding, if you don't have the right ecosystem for them or the right plants for them to feed, for example. So thinking about the restoration of ecological interaction is a bit, it's a big challenge, and I think it's really important if you want really to restore a whole ecosystem. Then of course you would have to identify which species, which are the partner species in the process that you are interested on. So if you're thinking about seed dispersal, then you are restoring plants, and then you need the animals there that are able to feed in those plants, and then are able to disperse them to safe sites. So if you want to restore, for example, you're going to miss. I'm not a specialist in restoration, but I'm just thinking loudly, actually. So it depends the stage of the restoration as well that you want to start with. If you want to start from the very degraded areas, or if you want to start from areas that already have some vegetation and you want to accelerate the recovery time. So you might need to introduce different types of species, and then do you have the animals there, or you also need to reintroduce the animals. So, and perhaps the landscape configuration is really important as well. So where are these restoration areas if they are surrounded by more conserved areas, for example, that you can have these in the network terminology, I'd say flow of information, but yeah, it's actually the movement, for example, of seeds by birds from one area to another. Do you have data for free, like the animals are there, you can do, or do you have to promote there? So these are very complex variables that you need to consider in these type of initiatives. So it just give you some brief ideas here. Is a lot to discuss in that way, but thank you for the question. It's really interesting. Thanks,
Corinne. I'm going to pass along to Arildo Diaz now, who is going to sort of synthesize, maybe, or help synthesize some of the discussion and the topics of the talk.
Yeah, thank you, Jean. Yeah, we have very few time to finish this section, but just to wrap up, I would like to thank you very much, all our speakers, for the great talkings, Karina, Robert, Adriana, also Jean, Ima, and in the background, the technique, Juan Pablo, and our audience for the fantastic questions. Just remember that we can continue the discussion, the community over. We had already opened a topic there about this section, so we can continue there. And I think with this, I sort of conclude the session and hope to see you after this short break. Thank you. Thank you very much.
Thank you for the invitation. Bye-bye.